All right, so good morning, guys. This is my biology catch-up class. We've been missing our our time together. I had to be in the hospital last week during homecoming, so I believe I missed one full class period with you, and then somewhat, especially with my honor class. The homecoming proceeding kind of take precedence, especially when it comes to getting floats and decorations and banners and stuff. So all that said, I wanted to revisit. We were transitioning from our cell project and all of our organelles that we discussed in the biology class, and we basically it was the beginning of chapter 5 that we were getting in, and there's some concept overlap that occurs when we talk about cell structures and the processes that they do. And one of the big conflicts that we run into at times is whether or not we should teach a particular content. That is, I should be doing a pretty good screen share right now. We talked about the hierarchy structure. We differentiated between eukaryotic, prokaryotic cells and animal and plant cells, like the plant cell out here, animal cells, right? Even about bacteria. Now, moving on, getting into the cell membrane brings us to really where I wanted to. So I'm going to go ahead and in this slideshow. And I want to talk with you guys, kind of like what I did with my 5th hour class on Tuesday before I lost contact with you because of the power outage in school. Basically, we wanted to carry on our unit in cells. We basically want to discuss how cells interact with their environment and how they take materials in and how they eliminate materials, and also how cells accomplish some pretty important life functions, specifically how they do balance. Right. So let's by focusing on cell membrane. Shift my picture off. All right, membranes themselves are incredibly important, can't be overstated. And oftentimes when we get into talk about biology, what we run into is it just becomes like the boundaries of the cell. Many of you guys talking through your cell projects said that it was like the protective layer of the cell. And that's true to a degree, but it doesn't do too much in the way of protection. The biggest thing is it separates the outside world from the inside world of cells. And the outside world and the inside world, you have to remember, are made of water. Okay. Another thing that cell membrane materials, they make up those membrane-bound organelles inside the cell. It's kind of weird to think about, but that's how it is. I mean, this same material works well to make little compartments that hold everything. Cell membrane itself, or smaller little compartments like mitochondrial powerhouse in the cell, or the Golgi apparatus, packages and transports and modifies proteins and such. So does the endoplasmic reticulum. Right. In fact, speaking of the endoplasmic reticulum, it's made up entirely of this stuff plus protein, and in some cases also ribosomes. And this material right here is incredibly important because it behaves kind of strangely. Right. Cell membranes and membranes in general are made up of 2 layers of these phospholipids. Now, if you look at my diagram on the right-hand side of the screen that I'm lassoing with my mouse, right? This really illustrates, okay? When you get down to the tiny atomic world, you're gonna have this region up here on the top, a polar head group and a phosphate, right? By the way, phosphate is charged, right? But so is this choline head right here. It's called acetylcholine, right? And it has this positive charge and it has a negative charge actually that balances with it on its phosphates. It's neutral, but it's electrical in its nature, unbalanced. Unbalanced in point. Water can attract to that very easily. Unlike down here, once you get past this little backbone, this glycerol backbone, all these fatty acid chains, these are all non-polar. They're hydrophobic. They repel water. They act like oil or fat sitting on top of and floating on top of water. Same thing with the cholesterol molecule. I don't really talk too much about, but they're in here. And what it is it just basically you'll have this layer of fat attracting fat on the inside. It's kind of like a plastic. And on the outside, it has these polar heads that stick to each other. So they kind of clamp to each other molecularly, like magnets sticking to each other, and it makes sheets. And if you put a bunch of that into water, what will happen is the fat sides will attract fat sides and the 2 layers of sheet spontaneously. If I had phosphatidylcholine, buy it, put it in a little jar, take a dropper of it, put it in water, and slip it on a microscope slide for you guys in class. Probably should do that. I don't know how much that costs, but it'd be a great demonstration to show you how this thing right here on the bottom right, this little spherical shaped kind of packet compartment that's spontaneously. Evolutionary biologists believe that this is evidence for how compartmentalization could spontaneously happen in nature, and that's how cells were formed. Somehow, a lot of these molecules were made and put together, then they would have stuck together. That could have been the first evolution of the cell. Trouble with that is that the environment that the evolution would happen in is incredibly unstable for these kinds of molecules. And they don't get just made spontaneously; they get made by very complicated enzyme structures. That's what we see in nature. I'm digressing a little bit about how biblical or naturalistic worldview. We as Christians give credit to God the Creator for making these things, right? And making predictable, orderly function systems in life, non-living nature. God's evidence of God's design displayed in what it does. And it goes through everything. It's a repeating theme in the Bible, and I'm trying to teach you guys what it's. Now I am kind of ranting and going on here. What do you need to take away? Right, this is going to lead us to transport. Right. You have polar heads, non-polar tails. They'll arrange in sheets that will ultimately make bubbles, and it does two very important functions. Like one, it makes compartments, so we can make basic cells like prokaryotic bacteria, and we can make organelles, eukaryotic cells, and the cell membrane themselves. Then the big thing, membrane structure will control material flow. Water can't just go through this, not easily. All right, it's very hard. Water environment separated, which is good. All right. We can do chemistry where we want to. We can compartmentalize stuff. Kind of like how you guys have a stomach that causes digestion. You don't want those contents throughout your body, with all the acids and such. Cells do the same thing. Now, before I get to transport, I just want to pay a little bit of a deference conversation about something that's very important for all cells. The book brings it up kind of like as a topic after a topic. So that's why I do it like this. That's homeostasis. Homeostasis basically means the balance of any system, the internal balance of the system and stability. Here's what I want you to think about in biology class. Homeostasis is what all living things do. We have an optimum condition for everything. Temperature. Your ideal temperature is 37 degrees centigrade or 98.6 Fahrenheit. And your doctor checks every time you go to go get a checkup. Get your vital signs taken at a hospital. Your blood pressure, how much pressure is on your blood vessel walls, and gas diffuse back and forth from your lungs and tissues and other organs. It needs to be 120/80. More about that in anatomy class. The amount of salt in your blood, the amount of oncotic pressure in your cells, all these things that you humans do it, but and you need to balance it. Plants balance, bacteria balance. Everything seeks this optimal point that was put there for its design. Homeostasis is how it's done. The cell membrane and organelles and cells in general function to maintain it. Right? In cells, we have a few terms that I'd like you to be familiar with. Okay, that homeostasis is a state of something called dynamic equilibrium. It's basically a constantly changing balance. All right. If you guys have ever tried to balance on maybe a balancing board, you're not perfectly still when you hit the balance point. You give little bits of input, positive and negative, to maintain general stability. Same thing happens molecularly with cells. Okay, various things can affect that homeostasis. So we're going to have changing conditions, and we're going to have mechanisms to help. What we're seeking with this dynamic equilibrium, this homeostasis, is an optimal range of existence. We want to stay right around our best points to make nutrition available, to provide energy for ourselves, get rest when we need it. And we work within an optimal range. And every organism has its own optimal range. We have standard temperature and conditions on Earth, considered like 25 degrees centigrade, 1 atmosphere of pressure, 21% of oxygen in the air. Those are like standard biological conditions, but that's what humans call that. But if you're a plant that goes to the desert, you have an optimal range different that grows in the tropics, right? Um, Humans even have different. Put me up in Nepal at the basic of Mount Everest, I would have a hard time for at least a month just kind of getting through my day. Breathing would be a little harder. But if you went and talked to the local Gurkha people that live in Nepal, they would be able to take it on their back and walk the mountain with you, right? Um, People can adapt. Everything can adapt. By the way, that brings me to what I'm talking about next: the ranges and tolerance. Okay, your ranges of tolerance basically for any condition, you have an upper limit and lower limit of handle. All right. And when you get outside of that, you start to have disease and distress. All right. Then ultimately you get to your limits of tolerance a bit further out. Um, when you exceed the limit of your tolerance as an organism, that means that particular change in state is probably going to explode. Here's an example. Um, I'm not really sure, to be honest with you. This little graph looks like a representation of a plant when I look at the optimal ranges and such. Okay, I could have it wrong. I have to research the textbook. Knowing what this one is doesn't matter as much as a particular organism. When you get it above 28 degrees centigrade, which is about 90 degrees, it kills it. Okay? When you get it around 22 degrees centigrade, the proteins that are in the cell become inactive in the cell. 18 degrees centigrade is its optimal point where everything works quite nicely for it. And as you get colder, get lower and lower, all right, maybe below 10, 12, 14 degrees centigrade. You get out of that optimal range, you get to 8 degrees centigrade, this organism's proteins become inactive. Kind of be like what would happen to you if you guys were getting hypothermia, all right? Then you get down to 2 degrees centigrade, and it kills the organism. This looks like something like a fungus to me. I could be wrong about it, but that doesn't matter. The emphasis was about the range of tolerance, the optimal range, the maximum range of tolerance. And know this, that this organism needs to have some means, some way in order to work where it doesn't get negatively affected. And maybe it's a type of animal that can move to go toward the optimal temperature places, find a niche there. Maybe it's a plant. Hopefully it's growing in an environment and it stays in the top range for most of the time, or when it's out. I digress. Okay. That said, homeostasis itself is maintained by these feedback loops. Right? We talk very extensively about this in the anatomy class. You guys, every organism has some kind of adaptation resulting in its design that can help it interact with its environment, make a change to itself to respond to a changing environment. All right. So that's what feedback loops are. We have a change in activity to keep an optimal condition. Okay? Positive feedback is more rare than negative feedback. So we'll just start with that and give you some case examples. We have a condition causes a response where the response increases. Right? The examples I put up here are blood clotting and childbirth. Right? If you cut your skin, right, you've ripped open blood vessels, bleeding out, fluid out of your body. That's a bad thing. Your body's response and senses that by the tearing of the blood vessels cells in there that will stick to those particular proteins in your blood vessels. And then they trigger the cells around them and stick to them, and it makes it clot. That increases until it makes a full platelet plug clot. That keeps you hopefully seriously. Doctors exploit that. You have really big cuts when they do stitches, and they stitch the wound closed and try to get the stuck together. That helps the healing happen. That just help with a little technology. Childbirth is another one. In a woman in her womb, basically at the bottom of her womb, the body of cells that go directly up to the spinal cord will connect to nerve cells that go straight up to the spinal cord to the brain. It's an area called the hypothalamus and the pituitary. Just giving you back, I don't have to have this memorized, but here's what happens. As the baby grows bigger inside mom, the baby can get too big, actually kill mom and the baby liver. The natural design that we put into reproduction and how that works. When that baby is developing, it gets to a point, usually around 6 to 8 pounds of weight, Baby's head's down, pushing on those cells. When it activates those cells, it sends a signal all the way up to the brain. The brain releases this hormone called oxytocin. The oxytocin goes out to the blood and down until it gets down to the muscles of the womb, uterus. I'm sure anybody in my ninth grade or other class, biology, whatever, you're familiar that with childbirth, a lot of cramping, pushing. That's what we're talking about. This is how it happens. And it gets greater and greater and more frequent until the point that the natural completed task needs to happen. The baby is birthed. And it's a miracle how it happens. But this is basically right. Negative feedback, on the other hand, this is where we negate it. Controlling the human body temperature is one of the best ways I can discuss it. Get really hot, your brain and some other sensors in your bloodstream pick up the change in temperature. They have thermoreceptors in it. They trigger you to start sweating throughout your body. Right? So start releasing that water. By the design, basically, water evaporates spontaneously into the atmosphere because there's always less in the air than there is right out of now. And it's a lot of heat in order to do that. So it spontaneously sucks the heat off of your body. That's why sweating helps you cool down, especially if it's breezy or if it's kind of arid and dry outside. The flip side of it, all right, you generate heat by burning energy. So when you get cold, you get to a point where your brain will start triggering your muscles to involuntarily contract and relax. And you shiver and tighten. All right. That'll cause your body to generate heat, balancing. Okay? Negative feedback does a balancing act. Positive feedback, we have a change that we have. I really, for the purpose of the course, I want you to understand as well. Homeostasis is what positive feedback and what negative feedback are, not memorize. Go through the course and talk about certain animal behavior. This will come back up to the course. Just introduce it as a concept. All right. Now moving forward, okay. I need to get into transport. Now there's kind of like we're talking about membranes, talking about a topic, and we just jumped and went into something unrelated. Textbook authors are good for that. That's basically where we are now. Right? When it comes to maintaining how cells work and interact with the environment, we have a condition. Basically, everything that happens in biology. I know we talked about some of these concepts about solutions before, about whether something is concentrated, solvent, that one the test from the previous quarter. Now we want to work this concept and actually apply them to cells themselves. The first thing that our book goes to talk about is solutions and moving things in and out of the cells. Okay. Since water is the universal solvent in cells, I believe they started here talking about simple diffusion and moving materials across the membrane, right? And they want to talk about the solvent first. So osmosis is the diffusion of movement through semi-permeable membrane. It's going to be water. And the membrane has to be very important. It allows certain substances to pass through, but not others. So some membranes do this, all right? They control flow, all right? Always a force working on that flow. This is going back to our conversation about, remember, things go from together, high concentration, or they're not, low concentration. It's a force just like gravity, just like electricity, positive and negative attracting each other. Right? It's kind of a fundamental one when it comes to materials in nature. Be balanced by other forces, but this is just how it works. Okay? We move things from high to low concentration. So that gives us the way of looking at different environments. We can have hypertonic solutions, isotonic solutions. I want you to look at those terms as being greater than or less than concentration. I know I'm sounding like a math teacher or a physics teacher like now, where I'm just kind of throwing terms around. Do that in science. We try to describe things and use a specific language. I want you to think about greater than, less than, or equal over school, right? Hypertonic solution is greater than or more concentrated outside of the or than the cytoplasm. Hypotonic, my outside, my environment out there would be less concentrated, rich inside. Isotonic would be balanced. Right? So here's an example of osmosis and hypertonic and hypotonic kind of playing out. Okay. I believe you have this diagram in your textbook. We have a semi-permeable membrane right here. All right. And what it's allowing to go through is only water. So what our experimenters do here in this particular image to illustrate is they take pure water on one side, all right, pour it in, and this membrane goes from side to side. So nothing can leak around it. Everything can go through, though, that can go. Then they put ocean water with solutes. They don't draw all the water molecules here in ocean water, and they set the timer at 0. Then after 2 minutes, what ends up happening is, because there's more solute matter dissolved on the right-hand side of the beaker, water is going to go from where it's higher concentration, more concentrated, where it's lower concentration. It's kind of weird to think about. But imagine this. I mean, this is all pure water on the left. There's some water and other stuff in here on the right. Water is less crowded over it, so the water will move and filter. And after 2 minutes, we start to notice a slight change in how much volume total is on either side of this the beaker. After 4 minutes or 4 hours, I don't know specifically, we have an equilibrium state. The attraction, the diffusion force, water moving from one side to the other, is balanced out by the force of gravity down on the higher water. And basically, once you get that balance, osmosis is taking place as much as it can. Now, I want you to apply this to what we talked about back in the first quarter. I remember showing you guys pictures involving diffusion. I did red blood cells, and we put red blood cells in salty water, red blood cells in pure water, and we put red blood cells in water that was balanced, like kind of you get at the hospital. What they do, so you don't stress your body tissue. That's it. Remember, like, water is going to move back and forth across this artificial membrane you see on the screen. It does the same thing through natural membranes, like the ones that you guys have in all of your cells. And you just change the environment, the water. The environment is changed by feedback, and the water moves. And that, in a nutshell, is hypertonic. Now, osmosis is a type of transport, all right? And we're going to get more general now. We'll talk about osmosis specifically because it's a solvent, all right? I think a physicist wrote that part of the textbook because we only really care about water in biology. But in fact, most biology classes, including AP, when we talk about osmosis, we only talk about water in definition. But I'm digressing again, all right? Passive transport is basically a movement of any molecule across the membrane without the use of any energy, all right? No chemical energy, no special stuff happening. They take on different forms, and there's a reason for that. It's because those phospholipid bilayers don't let things through easily. They regulate what can go. And some proteins and other structures. Big takeaway point right now: passive transport, no energy. Okay, so particles will move from areas of high concentration to low concentration. All right, just stuff I should have. Okay. 4 factors are going to determine, all right, whether a particle can go right across the membrane. All right, this is where we start kind of learning a little bit of molecular stuff, focusing on and expanding to everything. Okay. So, the first, particle size. I can't emphasize this stuff enough. This is a fundamental understanding, and then from here you can make sense of the rest of the transport and targeting and all these things, flashcards. All right. So, particle size. Extremely tiny particles tend to go through the membrane or they'll go through spaces more easily than transport proteins. Okay. particle size, like a good example of this is oxygen. Oxygen is a very tiny molecule, two oxygen atoms bonded together, one to the other. All right? They have an even distribution of charge, on negative end, balanced. So it's tiny, balanced in charge, non-polar. Which means oxygen is going to hit a cell membrane and just squeeze right through it. It's almost not. Same thing with carbon dioxide. Molecular formula is a little bit different. It's also tiny, it's also balanced. All right. Because of that, it will just move right through the cell membrane. So if you take that and you think about it, you might be reading into the fact I'm telling you basically how you guys breathe, correct? Or at least respiration. Yeah, you breathe in material into your body. You take in bulk amounts of it. Once those atoms and molecules get inside your cell, right, from there, what happens? You have to have gas transport. Diffusion makes it occur. Particle size is important. Particle polarity or charge. Particle shape is pretty important too. If it's big, if it's branched, it takes up a lot of area, it has huge two-dimensional geometry. If it's a whole cell, it's not going to go across the membranes. Okay, and then the composition of the membrane, right? If you have a polar molecule or a bulky molecule that needs to get across the membrane, all right, the only way it's going to get in is it's going to have to go through a protein. You don't have those proteins in your membrane that can carry that particular thing. It's not, including water. All right, so that plays out a lot in biology. It plays out a lot in cellular systems. Okay? So diffusion or diffusion and osmosis. Okay? Facilitated diffusion is a little bit different. Molecules are going to move through the cell membrane with the help of proteins. You'll notice the outside of the cell here, all right, the top area of my image, lots of these square-shaped molecules. Call them a sugar, all right? Lots of sugar outside, no sugar inside because the sugar is getting used, all right? From that perspective, I mean, you look at it, you need to get that stuff in. It would move in on its own. It can't get in because it's too big, so it just goes through a transport protein, all right? It responds to basically the concentration of the solution inside. Basic physics. Active transport. This case of movement, you require chemical energy to get it done. Okay, you can't move molecules across the membrane without it. So energy will be applied to the transport protein before the protein will be able to move across the membrane. There are a couple big examples of this: endocytosis, exocytosis. There's another type called the sodium potassium. I'm going to back out real quick. All right, stick with just the same structure that we saw. Add the complicated picture. All right, so an example of that. All right, sodium potassium pump. Picture cleans up a little bit. There we go. All right, so if you take a look here, all right, outside of my cell, I have a lot of sodium. All right, they don't really illustrate that very well. All right, and this particular type of cell, it's actually a nerve cell. It needs to pump sodium out. It has more inside, but there's a ton of it outside the cell, so it won't naturally get there. So how does it do its job? It takes ATP, the cell's universal energy molecule. A little bit more about that soon. Okay, it goes and it binds to that. The sodium will go into the protein because it attracts to it. ATP will go to that site, stick to it, and break, leaving a phosphate on there. That causes the membrane and protein to change. It'll kind of shift up and face outwards. All right, it'll drop the sodium. Potassium, there's plenty of it outside of the cell. It needs to come into the cell. So then it just kind of leaks over and diffuses down into its fit, and then the cell returns to its original shape and it drops the phosphate in it. That's basically how it works. Here's the thing. I don't want you guys trying to memorize how the sodium potassium pump works. Here's the big point, and what brings us back to what we were talking about. Okay. But basically the big picture is we we have too much sodium or a great amount of sodium outside of the cell, a smaller amount of sodium inside of the cell, but we still, for some reason, need out. We have the sodium going in, ATP energy being used by breaking, causing the protein flipping inside out. Sodium gets ejected, it's crowded, there's tons of sodium out there anyway. Potassium comes back, occupies the space, it's set the pump back to its original point. Okay, so potassium is going to build up inside the cell, and the phosphate's going to come back. And there's going to be some other way for getting the potassium out, like an unhealthy. But more on that later. I don't want to get too into all the physiology. Get back onto our original slideshow. So again, the big thing I was trying to show you, active transport requires chemical energy because we're concentrating, we're pumping stuff uphill, we're pumping ions or atoms to where there are lots of them already. Energy will be applied in order to use that protein. Now, some special cases of active transport are incredibly complicated in how they molecularly happen. They involve many proteins working together and make huge shape changes. This is where we get endo and exocytosis. Endocytosis is what happens when a cell will form a sac or something that touches the outside of the cell. It's off the vesicle. Remember vesicles from our organelle? Our organelle talk at the beginning of the chapter, the project. Vesicles and vacuoles, small little cell membrane. They get made. Cells eat big things from the outside world, receive material inside. Exocytosis, usually the opposite of that. I just want to show you what it looks like right here. Right? Now, the cell membrane, the outside of the cell, the inside of the cell, large particles come in contact with the cell membrane. Usually there will be a receptor protein that triggers this process, but I don't want to get too complicated. The big thing, endocytosis, the cell, big particles come in. There are going to be a lot of motor proteins kind of forcing this pinching. This doesn't happen spontaneously. It's the result of things being triggered. And it squeezes the vesicle in until a point that it pops out. All right. Now, here's the big thing to think about. How does this happen so easily? Well, one, this doesn't. Endocytosis part doesn't. But if I'm trying to eject materials out, and if I come from maybe the Golgi apparatus, all right, and I had a cell membrane phospholipid bound packet of materials, protein maybe, all right, when those lipids bump into the cell membrane lipids, they kind of merge into it. They're soluble in it. And it just basically opens up and forms a pocket cavity, allowing the ejection of whatever was inside. I'll tell you this, cells don't typically take stuff in from the outside and then just spit it right back out from the inside. They bring in the materials that they need for working the cell. It's actually a guided activity. But this is just generally a good figure to show you what it looks like. Do either. So from there, review questions that we have that they put up here, they're kind of generalized. They connect to your reading. I don't want to take time too much to go over it. All right. But this should basically conclude our chapter. So what I'm going to do for you now, I'm going to go ahead, back out the slideshow. I'm going to put a pause where I'm going over today. I also want to show you a couple of really good videos too. All right. Some of them are already posted here. Okay. Cell structure, if you need a review of what different organelles do, we watched this in class. I'm about 100% sure on that. All right. And then cell transport overview. This basically is a short animation kind of based review. But if you've never heard the concept, significance, or example, give a little bit kind of exhausted, and excuse me, a little bit too brief. My apologies. All right. Now it said my embed code doesn't work right. What you can do is click on the watch YouTube location up on the YouTube app, right, on your student iPad. Then I have some overview, osmosis, basically described using dialysis. I'm going to show you how this happens in class. All right, and then just some experiments, just do it that way. have you. Right? Additionally, I want to guide you to YouTube because it's some really good videos on there. Not this one. Let's do a different search because I'm junk on that. All right. You guys know that I'm kind of a fan of the videos and animations. Do really simple. I would love for you to watch the side of the cell membrane video. Also the cell transport video, right? Then look up the Homeostasis channel on YouTube. You're gonna have a video list. They'll have a cell transport kind of rundown. Review any of those concepts we got done talking about. Back down to introduction to cells. All right. Those are really great. And my 5th hour students, well, we watch them together. That said, I'm going to go ahead and stop the screen. And this could be a good video for you guys in the future. Getting ready for the next test. That said, we'll I'm going to take a break right now and welcome back. But I'm basically going to end the recording from this point, switch over and chapter 6 video, and I'm going to talk with you guys about ATP, what it is, how important it is. And then I'm also going to focus on transcription, translation. Well, really what I'm going to focus on DNA replication. I'm going to pay deference to those topics that are posted in chapter 6, but as you in the classroom. So today being an A day, 3rd period is going to be a class that's going to kind of on what I'm going to post online for the next video. Okay. My B-Day students are on they're going to be able to emphasize this stuff in chapter 6. My honors bio students on Tuesday, watching the Amoeba Sisters videos, the lecture that I just gave you right now, basically what I 5th hour. So the only way you're behind is one of the good things about these videos is they can pause you to death and you need to get up along the way instead of putting your head on the desk. Anyhow, I'll see you.